Hello and welcome to this Bio 6 lecture on water. Now, you know what the definition of a molecule is. A molecule is a particle that's made out of atoms joined together. Well, starting with this lecture, we're going to start talking about some of the important molecules that the human body is built out of. And one of the most important and the most abundant molecule that the human body has are water molecules. And so that's going to be the focus of this lecture is water molecules. So as you can see from the molecular structure here of the water molecule, each water molecule is made out of one oxygen atom and two hydrogen atoms with the covalent bonds shown by the red lines in this figure. And incidentally, notice that the water molecule sort of has a V shape with the oxygen atom at the point of the V. That is actually the shape of water molecules, that particular shape that you see there. And so yes, water molecules are the most abundant molecule in the body by far. In other words, there are more water molecules in your body than there are protein molecules or DNA molecules or fat molecules or you name it. Our bodies are roughly half water by weight. And, a good way to underscore the importance of water to the proper functioning of our bodies is this. You can only go a few days without taking in any liquids, any watery liquids, and then you would quickly get ill and die if you didn't take any more. Whereas with foods, you could go weeks without eating if you have to, but water is just more important. It's just a few days without water and you become ill and non-functional. So why is water so important to the proper functioning of our body? Well, water has several properties that are essential to keeping our bodies functioning properly. And the lecture outline lists two of these important properties of water. One, water dissolves many substances, and two, water cools as it evaporates. And so we're going to talk about these two important properties of water to keeping our bodies functioning properly one at a time, starting with water dissolving many substances. And so we know that water dissolves a lot of things just from our everyday experiences around the house. You know that salts dissolve well in water, sugars dissolve well in water. Most proteins dissolve pretty well in water also. If you think of milk, milk is a watery liquid that has a lot of proteins dissolved in it. So yeah, water dissolves many substances very well. And the examples that I just named, salts and sugars and proteins, we can think of those as nutrients for the body, right? Those are substances that your body uses as nutrients. So we can say that your body dissolves, we can say that water dissolves nutrient substances very well. And that's important to the proper functioning of your body because it helps your body transport nutrients throughout the entire body. And what I mean by that is this. Your blood is a watery liquid. And so thanks to the fact that your blood is mostly water, that allows your blood to dissolve, nutrients like salts and sugars and proteins. And once those are dissolved in your blood, it's easy for your body to transport them because all your body has to do is to circulate the blood throughout the body. And that way, all the cells and tissues and organs of your body can get those nutrients that are dissolved in the blood. Okay, yeah, so that's one of the main importances of water to the body. It dissolves many substances like those nutrients that I just named, and that helps your body distribute those substances throughout the body. Well, what we're going to do now is focus a little bit more on water's ability to dissolve substances. Roughly speaking, we can subcategorize all substances into hydrophilic substances and hydrophobic substances. And here in a second I'll give the actual definitions of those terms, but just in a nutshell, briefly, hydrophilic substances are substances that like water, and hydrophobic substances are substances that don't like water. And so let's now focus a little bit more on those terms one at a time. Hydrophilic substances. So the term hydrophilic literally means water-loving. And so those are substances that like water. They are attracted to water and water is attracted to them. And one of the characteristics of hydrophilic substances is that they dissolve well in water. And so if something dissolves well in water, it's a hydrophilic substance. It dissolves well because it likes the water and water likes it. Some good examples of hydrophilic substances are salts. For instance, you know from experience that salt dissolves very well in water. Well, one of the reasons that salts dissolve very well in water is that salts are made out of ions. Remember that ions are electrically charged atoms and molecules. And each crystal of table salt is actually made out of ions. Each crystal of table salt is made out of sodium ions and chloride ions. And so what I'm saying is any substance like table salt that is made out of ions is always going to be a hydrophilic substance. That's why salts dissolve very well in water. They're made out of ions, so they're hydrophilic. In the web handout for this lecture, in addition to ionic substances like salts, the web handout tells you that carbohydrates are also hydrophilic substances. Now, I haven't talked about carbohydrates as molecules yet. I haven't really defined what they are. But just as a preview, sugars are a type of carbohydrate, and we know that sugars dissolve very well in water, right? So it's not a stretch of the imagination to say that carbohydrates are hydrophilic molecules. And the web handout also lists as another example of hydrophilic molecules, proteins. And we know that from experience, like I mentioned a few moments ago, milk is a very high protein liquid, and those proteins dissolve in the milk because milk is mostly water, and so water can dissolve most proteins very well. And I know the next thing I'm going to say sounds a little strange, but water itself is a hydrophilic substance. Water dissolves very well in water, right? So water molecules themselves count as a hydrophilic substance. So just to summarize so far, hydrophilic substances are substances that are attracted to water and water is attracted to them. And as a consequence of that, we can also say hydrophilic substances are the molecules of hydrophilic substances dissolve well in water. Oh, incidentally, although when we're talking about hydrophilic molecules, we usually just call them hydrophilic molecules, they can also sometimes be called polar molecules. Polar means the same as hydrophilic. And some people use the word lipophobic molecules to mean the same thing as hydrophilic molecules. I usually don't use that term, but you should be aware of it. Okay, so that's what hydrophilic molecules are. They dissolve well in water. They're attracted to water, and water is attracted to them. Matter of fact, the term hydrophilic means that literally. Hydro means water, and philic means loving. The other term now, hydrophobic, hydro means water. Phobic means fearing or hating. And so these are molecules that don't like water, and water does not like them. Hydrophobic molecules. And so in the web handout, I said that, well, these are the molecules that don't dissolve well in water. And so if you think about, well, what sort of molecules are you familiar with that are hydrophobic, that don't dissolve well in water? Oil is a good example. We know that oil and water don't mix. Here is me pouring some cooking oil into a glass of water, and that oil is never going to dissolve in the water, right? It forms an entirely separate layer, and so that shows that it's not, oil is hydrophobic. It's not dissolving in water. In addition to oils, another good example of hydrophobic molecules are fats. And some common fats that we have around the kitchen: butter. Butter is milk fat, as is cheese. Cheese is also primarily made out of milk fat. Cooking lard is also a type of fat. And none of these substances dissolve very well in water, so they are all hydrophobic substances. And I don't have a picture of it here, but waxes are also hydrophobic substances. Waxes don't dissolve in water. Waxes even seem to repel water. Okay, now I want to just mention something here about how you might recognize a hydrophobic molecule if I show you its molecular structure. The next slide will show a picture of a typical fat molecule. Here it is. So you can see fats are rather large molecules, but what I want to point out to you here is this. Notice that large regions of the fat molecule are made out of only carbons and hydrogens, like this region here and this region here and this region here, the fat molecule. It's just carbon atoms and hydrogen atoms. No oxygen atoms in these regions, no nitrogen atoms, just carbons and hydrogens. And so that's a trait that all hydrophobic molecules have. If you see a molecular structure and large regions of the molecule are just made out of carbons and hydrogens, you should assume that that molecule is going to be a hydrophobic molecule. Okay, now that we have introduced the concepts of hydrophobic molecules and hydrophilic molecules, it's time to move on to the next concept, which is called the like mixes with like principle. And what this essentially means is that hydrophobic things want to mix with other hydrophobic things, and hydrophilic things want to mix with other hydrophilic things. Each kind wants to mix with its own kind. It doesn't want to mix with the opposite kind. So, one aspect of this like mixes with like principle is this: hydrophilic molecules mix with other hydrophilic molecules, not with hydrophobic molecules. And just to give an example of that, remember that water is a hydrophilic molecule, and ions, like salts, are also hydrophilic molecules. And we know that water mixes with salts. Salts dissolve very well in water. And likewise, carbohydrates are also a hydrophilic molecule, as are salts. So salts and carbohydrates are going to mix well with each other and be attracted to each other. But water and other hydrophilic molecules are not going to be attracted to hydrophobic molecules like waxes and oils and fats. And another instance of this like mixes with like principle is this for hydrophobic molecules. Hydrophobic molecules mix with other hydrophobic molecules, not with hydrophilic molecules. So fats and oils will mix very well with each other and dissolve each other, but hydrophobic molecules like oils don't want to mix with hydrophilic molecules like water or sugars or proteins and things like that. So again, this like mixes with like principle basically says each kind, hydrophobic or hydrophilic, wants to mix with its own kind, and it does not want to mix with the opposite kind. Okay, so let me briefly summarize what we've learned so far in this section. All substances can be subcategorized into hydrophilic substances or hydrophobic substances. Hydrophilic substances like water, they dissolve well in water. Hydrophobic substances don't like water, they do not dissolve well in water. And each of these kinds wants to stick with its own kind. Hydrophilic substances like water and carbohydrates and proteins and salts want to be together with each other. And hydrophobic substances like fats and oils and waxes are attracted to each other. They don't want to mix with hydrophilic substances. And another concept that we learned is that most nutrients in the body, like salts and carbohydrates and proteins, for example, are hydrophilic substances. And because those are substances that dissolve well in water or watery fluids, like the blood, the body uses that ability of water to dissolve many molecules, many hydrophilic molecules. The body uses that to help distribute nutrients throughout the body, because those nutrient molecules, being hydrophilic, dissolve well in the blood, and then the blood can transport those nutrients all throughout the body when the blood circulates. Okay, so that's one important property of water to the proper functioning of the body, is that water dissolves many substances. Another important property of water to the proper functioning of the body is that water cools as it evaporates. And so your body uses that to help you maintain temperature homeostasis. In other words, your body uses the evaporation of water through sweating to help keep you in the proper temperature range for the human body. And so if your body senses that you are going above the homeostatic proper range for body temperature, the body will activate your sweat glands, which are the effectors for cooling your body temperature. And so, your sweat glands put some of your body water on the surface of your body, and that water then starts evaporating. And it's a natural property of water that when it evaporates, it lowers the temperature. So cooling your body is also an important property of water for the body. The next topic in the lecture outline is the term dissolve. So imagine that you wanted to dissolve some salt into some water. Well, you know what that would look like, right? You put some salt crystals maybe in a spoon and then put them in the water, and the salt crystals seem to disappear. But obviously the salt has not vanished, because if you taste the water, you can still taste the salt, so it's still there. Well, when we say that you've dissolved that salt in the water, what that really means is the molecules of the salt have become evenly spread out in the water. And so that's the definition of dissolve. It's when the molecules of a substance have become evenly spread out in the liquid. And incidentally, that's why the salt crystals seem to disappear. They get spread out so thinly in the glass of water that you just can't see them anymore. Well, there are some special terms that relates to dissolving substances. And this next illustration shows them. There's the salt being dissolved into the beaker of water. So one of these three terms that relate to dissolving is solute. Solute just means the substance that is being dissolved, the substance that gets dissolved. So in this example, the salt is the solute. The term solvent means the liquid that does the dissolving. So in this example, water is the solvent. And the term solution means the liquid that you get when you've dissolved the solute in the solvent. In other words, the salt water that you would end up with is the solution. You would say that you've made a salt solution. And so let me go over these three terms again, but using sugar. If you poured sugar into a glass of water, the sugar molecules are the solute, the water is the solvent, and the sugar water you get is the solution. We're going to be using these three terms quite often throughout the semester, so please get used to them. And unfortunately, you notice that they all sort of look alike, and so try to keep them distinct in your mind. The solute means the molecules that get dissolved. The solvent is the liquid that does the dissolving, and the solution is the liquid with the solute dissolved in the solvent. Now, incidentally, we're going to use of these three terms, probably the word solute is the one that will come up the most. And so throughout this entire semester, if I say solute, just think of some molecule that's been dissolved, typically dissolved in water. Just means a dissolved molecule. And throughout this semester, when we talk about solvents, almost always water is going to be the solvent. And here's why. This is a biology class, and in living things, water is always the solvent. And what I mean by that is, if you think of your body fluids, they are all water-based, and so water is the solvent for those fluids. Your blood is a watery fluid. The liquid that fills the inside of your cells is a watery fluid. The fluid that you find between the cells of your tissues, your tissue fluid, is a watery fluid. So in all your body fluids, water is the solvent. Okay, so just to recap, the term dissolve means when molecules become evenly spread out in a liquid. Now, they're not initially evenly spread out, right? If you pour that salt into the liquid, into the water, initially the salt molecules would be all together, and then they have to spread themselves out before they can be dissolved. Well, there's a term for the process of solute molecules spreading themselves out in a liquid, and that term is diffusion. Diffusion essentially means the process of those solute molecules spreading themselves out in the liquid so that they can become dissolved. And to illustrate diffusion, rather than using the salt here, I'm going to use some green solute molecules. So imagine you had a dropper full of some sort of green ink, and you had a beaker of water. And let's say that you put that drop of green ink into the water. And so that's what you see right here in this figure. Well, so it's not dissolved initially, because dissolve means that the solutes have become evenly spread out, and they're not yet. But given a few minutes, they're going to start to spread themselves out, right? So what I'm saying is that process of the solutes spreading themselves out is what diffusion means. And if you look at the definition I gave for diffusion in the lecture outline, it says the movement of solutes from an area of their high concentration to an area of their low concentration. And if you think about it, that's just a fancy way of saying spreading out, because when that drop of green ink first hits the water, like you see right here, all those green solute molecules are all concentrated together right here. So that's a high concentration area. And the surrounding areas right there are, well, they're pure water, so they are very low concentration of the green solute molecules, right? So when we say the solutes are spreading, they are moving from that high concentration area to that low concentration area. And that's actually the technical definition of what diffusion is: movement of solutes from high concentration to areas of low concentration. And obviously the solutes are still diffusing even over here, right? Because here they are at a higher concentration than they are in these areas right here, and so they're going to continue to diffuse as long as there's a high concentration area and a low concentration area. Yeah, given time, even this this arrangement right here is going to continue to diffuse as those solutes continue to move from their high concentration area to their low concentration area. So that's what diffusion is: movement of solutes from their high concentration area to their low concentration area. And they keep diffusing until they are completely evenly spread out, at which point you say that the solutes are dissolved. Okay, what we're going to do next is review the definition of diffusion a second time, but we're going to review it while zooming in on this group of green solute molecules right here. So when we first put the drop of green ink into the water, all those green solute molecules were concentrated together right here, and surrounding them were areas of the water where those green ink molecules were at a low concentration, right? Because it's pure water, so there's essentially no green ink molecules there. So let's zoom in right there on the green ink molecules. Here they are. They're all concentrated together there, so they are at a high concentration there. And here's the surrounding water where those green ink molecules are at a low concentration. And again, the definition of diffusion is solute molecules moving from their area of high concentration to areas of their low concentration. And again, it's just a fancy way of saying solute molecules spreading out. And yes, they continue to diffuse until they have become evenly spread out in the beaker of water. At that point, we say they are dissolved, because dissolved means when the solutes have become evenly spread out. Okay, very good. So I've been using green ink solute molecules as my example diffusing molecules in these examples, but it's not just green ink molecules that diffuse. All solute molecules naturally want to diffuse. So it doesn't matter if we have used green ink molecules or salts or carbohydrates or proteins or any other solute molecule. All solute molecules naturally want to diffuse. They all naturally want to go from their high concentration area to their low concentration area. And just as kind of an analogy to this, think of things like rocks on a hillside. They want to naturally go from their high elevation area to low elevation areas, right? That's the natural direction that rocks on a hillside or other objects want to go. They want to go from high elevation to low elevation. They do not spontaneously want to go from low elevation to high elevation, right? You never see a boulder just sitting down here suddenly rolling on its own up to the top of the hill. That's not its natural direction of travel. Its natural direction is from high elevation to low elevation. So I'm saying in an analogous way, the natural direction of solute molecules is from high concentration to low concentration areas. The natural direction is from where the solute molecules are high concentration here into the surrounding low concentration areas there. And the reason I'm making a big deal of that is it's going to become an important concept in some other lectures later on in this class. So what I'm saying is anytime you see a high concentration of a solute right next to a low concentration of a solute, you should say, okay, that solute's just going to naturally want to go from the high concentration area to the low concentration area. That's its natural direction of movement, just like rocks naturally want to go from high elevation to low elevation. Okay, well, imagine that we repeat this diffusion experiment that you're seeing here, but we're going to make a slight change in it. Instead of just having the beaker filled with water and only water, we're going to put a cell in the beaker. Here's a cell. Well, when we put the drop of green ink into the water, the water with the cell in it, the green ink molecules would still diffuse, but they would not be able to enter the cell. So you'd get something like this. And why can't they get inside the cell? Well, it's because of the cell membrane. Remember, the outer layer of each cell, the outer boundary of each cell is called the cell membrane, and cell membranes are barriers that prevent most solutes from diffusing through them. I think the next slide is a close-up. Imagine we're going to zoom in on the cell right here. There it is. And here's those green solute molecules right there. Well, yeah, the green ink molecules can still dissolve in the solution outside the cell, but cell membranes are just natural barriers to most solutes. So the cell membrane blocks those solutes from diffusing through, and well, that's why those green solutes would not be able to diffuse through the membrane to the inside of the cell. Now, how exactly is the membrane able to be a barrier to block solutes from diffusing? Well, to answer that question, let's imagine we're going to zoom in even more. We're going to zoom in right on the membrane there. Here it is. We haven't yet talked about what the molecules of cell membranes are. In other words, we have not yet talked about what types of molecules make up cell membranes. But let me give you a preview. Cell membranes are made out of a type of molecule called a phospholipid. And you see them here. Each of those little orange balls with the two tails hanging down is a phospholipid molecule. So, yeah, we'll talk about this more later on in the course, but cell membranes are made out of a type of molecule called phospholipids. And here you see the cell membrane, those phospholipids being a barrier that's stopping those solute molecules from diffusing through. But what I was going to answer is, well, how is it that the cell membrane with its phospholipids can be a barrier that stops those solutes from diffusing through? And here's the answer. It relates to the like mixes with like principle that we talked about earlier. Those phospholipid molecules, here's a picture of one, are hydrophobic. Phospholipids are related to fat molecules. And remember that fats are hydrophobic molecules. So the cell membrane is hydrophobic because it's made out of these hydrophobic phospholipids. But most solutes are hydrophilic. Now, why do I say most solutes are hydrophilic? Well, we're talking about the solutes inside the human body. And the human body, remember, for all your body fluids, water is the solvent. And so that's naturally the type of solutes that we expect to find in the human body are hydrophilic solutes, right? So the most solutes are hydrophilic. The membrane is hydrophobic. And so for those solute molecules to diffuse into the cell, those solute molecules would have to pass through the membrane, right? And the membrane is hydrophobic, so those hydrophilic molecules would have to mix with the hydrophobic membrane. And remember, the like mixes with like principle tells us that hydrophilic molecules don't want to mix with hydrophobic molecules. And so that's the secret of how the membrane stops most solutes from diffusing. Membrane's hydrophobic, most solutes are hydrophilic, and so those hydrophilic solutes don't want to mix with the membrane to pass through it. Okay, so to just review, the cell membrane is hydrophobic, and that's what stops most solutes from diffusing through the cell membrane because those most solutes are hydrophilic. Okay, let's kind of zoom out again to this view of the cell. So those solutes generally are not able to diffuse across cell membranes because the solutes are hydrophilic and the cell membrane is hydrophobic. But there's an interesting exception to that rule. Water molecules can pass easily through cell membranes. And the reason that's kind of interesting is water is hydrophilic, right? Water is attracted to water. Water dissolves well in water. So water molecules are hydrophilic. So it seems really strange that water is able to pass through the hydrophobic cell membrane, and yet it is so. So in general, the hydrophobicity of the cell membrane stops hydrophilic solutes from diffusing through, but water is the exception. Water molecules move very easily through cell membranes. Okay, keep that fact in your mind that water can move easily through cell membranes because it's going to become important here in just a second when we talk about something called osmosis. All right, so let's begin discussing osmosis. So imagine we're going to do an experiment. We take a cell, and just all cells have some solutes inside the cell. Now, for today's lecture, don't worry about what types of solutes those are. If you're curious, you find salts inside cells, that means ions. You find carbohydrates, you find enzymes and other proteins inside cells. So all cells have some solutes inside them. Okay, now we're going to take this in this thought experiment that we're doing. We're going to take this cell and we're going to put it into a beaker of pure water, like you see here. So, to begin with, we know that those solutes want to diffuse out, right? Because you know that the natural direction of solute molecules is to move from areas where they are in a high concentration, like in this case inside the cell, to areas where they are at a lower concentration, which in this case is outside of the cell. Now, I should introduce a term here. So look at the solution outside the cell, which in this example is pure water. So it has a lower solute concentration than the cell does, right? Because the cell has some solutes and the water has no solutes. So yeah, the solution outside the cell has a lower solute concentration than the cell does. And a term for any solution like that is a hypotonic solution. Hypo means less than or beneath, and tonic you can think of as meaning the solute concentration of a cell. So any solution that has a lower solute concentration than a cell does is a hypotonic solution. It doesn't necessarily have to be pure water. As long as the solution has a lower solute concentration than the cell does, we call that solution a hypotonic solution. Anyway, so here you see this cell in this beaker of pure water, which is a hypotonic solution. And as I was saying, there's a higher solute concentration inside the cell than out. So the natural direction of those solutes is they want to diffuse out of the cell, right? All solutes want to move from their high concentration area to their low concentration area. Do you think those solutes will be able to diffuse out of the cell? And the answer to that question is no, because you know that cell membranes are barriers that block most solutes from diffusing. So those solutes will not be able to escape the cell and go out into the water. The membrane blocks them, you know that. Okay, so let's freeze those solutes there for a second. So those solutes can't cross the cell membrane, but water molecules can cross the cell membrane. And if you did this experiment, what you would see is this. The water molecules would be moving into the cell. And what you're seeing here is an example of something called osmosis. Osmosis is defined as the movement of water molecules across a cell membrane, and the water always moves towards whichever side has the higher solute concentration. And so what I mean is, is the water moving inside the cell or coming out of the cell? In this example, the osmosis is the water moving inside the cell. And why is the water moving inside the cell? Well, the short answer is solutes attract water molecules. And so think of solutes as essentially being like little magnets that attract water molecules. And so that's why the water is moving in this osmosis example into the cell, because that's where the higher solute concentration is. So wherever there's a higher solute concentration, inside the cell versus outside the cell, that's where the water is going to move. And in your lecture outline, I summed it up, but in a few words, I said water moves towards the solutes. That's kind of the basis idea behind osmosis. Okay, so anyway, so yeah, what you're seeing here is an example of osmosis. It's water moving across a cell membrane, and that movement is always the water moving towards the higher solute concentration, which in this example is inside the cell. Okay, so what's going to happen to that cell? Well, if you ever played with water balloons, you know that the more water you add to a water balloon, it stretches to a bigger size. And so that's going to happen to this cell. It's going to take on water and grow larger and larger. Now, the cell may burst. The official science name for bursting is lysis, or we say that the cell will lyse. And so, yeah, if you put a cell in a hypotonic solution, the cell is going to gain water and enlarge, and the cell may lyse. Now, the cell won't necessarily lyse. It depends on what type of cell it is and how hypotonic the solution is and how long the cell was in the hypotonic solution. The cell will definitely gain water in a hypotonic solution and enlarge, and it may or may not lyse. It may or may not burst. Okay, now we are going to try a different thought experiment. We're still going to use the cell and the beaker, but the solution outside the beaker is no longer going to be hypotonic. So here's the new experiment. We've got our cell right here, and those are the solutes inside the cell that you see there. But instead of having a hypotonic solution outside, we're going to have a solution that actually has a higher solute concentration than the cell, right? You can see here that there are more solutes outside than inside. Well, any solution like that that has a higher solute concentration than a cell has is called a hypertonic solution. Hyper means above or greater than, and tonic again you can think of as meaning the cell solute concentration. So a hypertonic solution is any solution that has a higher solute concentration than a cell. Okay, so here's my question to you. Where do you think the water is going to move, into the cell or out of the cell? And remember that the golden rule of osmosis is water moves towards the higher solute concentration because solutes attract water. Well, as you might have guessed, in this situation, by osmosis, the water will move out of the cell because, again, solutes attract water molecules. So whichever side of the cell, the inside or the outside, has the higher solute concentration, that's where the water is going to move, towards the higher solute concentration. Yeah, so anytime you put a cell in a hypertonic solution, the cell is going to lose water by osmosis. And notice that it's still osmosis. It's still water moving across the cell's membrane towards the higher solute concentration side. But unlike our previous experiment with the hypotonic solution, when we use a hypertonic solution, the water comes out of the cell, not into the cell. All right, yeah, so cells will always lose water by osmosis when you put that cell in a hypertonic solution. Okay, so what's going to happen to the cell? Well, I think a good analogy in this situation is grapes and raisins. You know that raisins come from grapes, and grapes start off as nice kind of round things like you see this cell right here. But when they take grapes and they evaporate the water out of them by putting them in the sun, the grape gets smaller and it gets all shriveled and turns into a raisin as it loses water. And so that's exactly what happens to cells when you put them in a hypertonic solution. They lose water, and so then they shrink, but they also get sort of crinkly and shriveled like a raisin. And there's an official name for being crinkled in science words. It's called crenated. So we say that when you put a cell in a hypertonic solution, the cell loses water and gets smaller, it shrinks, and the cell becomes crenated. All right, and so let's do one more osmosis thought experiment. We're still going to put the cell in a beaker of solution. Here we go. But notice the solution, oh yes, again, so those three are the solutes inside the cell. So notice in this situation, the solution outside the cell has the exact same solute concentration as the cell. Well, any solution like that, that has the exact same solute concentration as the cell has inside, is called an isotonic solution. Iso means equal to, so the term means equal solute concentration as the cell. Okay, so here's my question to you. Where do you think the water is going to move, if anywhere? And the answer is nowhere, because there's an equal solute concentration inside the cell and outside. Because those two solute concentrations are equal, there's no, greater force on the water either pulling it inside or outside. The water molecules are pulled equally in or out, so that essentially the water molecules just don't move. They're being pulled equally into the cell and out of the cell because there's equal solute concentration. Yes, so in an isotonic solution, cells stay the same size. They don't gain or lose water, so the cell stays the exact same size. Okay, so now you know the osmosis, it's movement of water across a cell membrane, and the golden rule of osmosis is the water always moves towards wherever there's the higher solute concentration, either inside the cell or outside the cell. And you know that the solutions outside the cell we can describe as hypotonic, hypertonic, or isotonic, depending how their solute concentrations compare to those of the cell. Okay, good. Now let me give you a drill here. So what we see down here are some red blood cells. And for each of the cells, I want you to try to tell me whether that cell was soaked in a hypotonic solution, a hypertonic solution, or an isotonic solution. And let's start off with this cell right here. And I'll just tell you right here, looking at this, this is the normal, natural shape of a red blood cell that you're seeing right here. And so, do you think that cell, that red blood cell there, has its normal, natural shape? Was it soaked in an isotonic solution, a hypotonic solution, or a hypertonic solution? And the answer is isotonic solution. Because remember, cells keep their own natural size and shape when you put them in an isotonic solution because they don't gain or lose water. Okay, and so over here, let's do this cell. This cell looks like, compared to the normal red blood cell, this cell is shrunken and crenated. Remember, crenated means shriveled like a raisin. So was this cell soaked in an isotonic solution, a hypotonic solution, or a hypertonic solution? And the answer there is it was soaked in a hypertonic solution, because it's hypertonic solutions where water exits the cell and the cell gets all smaller and crenated. And what about this cell right here? Here's this red blood cell. Looks like it's larger and swollen up compared to a normal red blood cell. So what sort of solution was this cell soaked in? And the answer is a hypotonic solution, because remember, it's hypotonic solutions. When you put a cell in a hypotonic solution, the cell gains water and swells up to a larger size, and the cell may lyse, the cell may burst, depending on the exactly how hypotonic the solution was. Okay, very good. Now, I think you'll agree that the cells of your body don't want to become all shriveled and crenated and shrunken. That wouldn't be healthy for them. And likewise, the cells of your body don't want to get stretched to a larger size and risk lysing. The cells in your body want to keep their own normal, natural shapes. So don't be afraid to be obvious when I ask you this question. Do you think your body fluids are isotonic, hypertonic, or hypotonic? And so the answer is isotonic. Your blood and tissue fluids and other body fluids are all isotonic, because that allows your cells to keep their own normal, natural shape, which is the healthiest shape for your cells. Yeah, so just repeating, your blood is isotonic to your cells, and your tissue fluids are isotonic to your cells. All your body fluids are isotonic to your cells. Okay, now, so the body's own natural body fluids are isotonic to our cells. But when we go inside the hospital, sometimes they give us new body fluids. An IV solution, an intravenous solution, is a hospital solution that they put into our body. So they're giving us new body fluids. So do you think when they give you an IV solution in a hospital, the hospital IV solution, do you think it's isotonic, hypertonic, or hypotonic? And the answer is isotonic. Almost all hospital solutions are isotonic for the same reasons we just talked about. They don't want to give you a hypertonic IV solution usually, because that would cause your cells to shrivel and crenate, and they don't want to give you a hypotonic IV solutions in the hospital, because when those got inside you, they would cause your cells to enlarge and maybe lyse. So almost all hospital solutions are isotonic solutions. Okay, so let me finish off this section on osmosis by giving you some drill questions here. All right, so here is a cell, and as usual, it has a certain concentration of solutes inside. And let's imagine that we're going to put this cell in a solution that has a higher solute concentration than the cell, like you see here. Okay, so my first question is this solution outside the cell an isotonic solution, a hypertonic solution, or a hypotonic solution? And the answer is a hypertonic solution, because those are solutions that have a higher solute concentration than a cell has. All right, and then my next question is, will the water move in or out of the cell in this situation you see right here? And the answer is the water would exit the cell, because, well, the idea is that water always moves towards the higher solute concentration, which in this situation, where the solution outside the cell is a hypertonic solution, that's the higher solute concentration outside the cell. Good, okay, and my next question is, what's the name of this process where water moves across a cell membrane towards the higher solute concentration? And of course, the answer is osmosis. That's defined as the movement of water across a cell membrane towards the higher solute concentration. And my last question is this: what's going to happen to the cell as it's losing water in terms of the cell shape? Well, the answer is the cell will shrink and get crenated. Crenated means where the cell gets all folded up like you see there, like a shriveled like a raisin. Okay, good. So I think you have understood hopefully the concepts of osmosis and hypertonic and hypotonic and isotonic solutions. Now, unfortunately, the terms hypertonic and hypotonic sound kind of similar to each other, but they mean different things. One is a solution that has a higher solute concentration than a cell, and the other is a solution that has a lower solute concentration than a cell. So try to keep hypertonic and hypotonic distinct terms in your mind. Okay, the next topic on our lecture outline is acids. And we're going to first talk about, well, what is the definition of an acid? What does that word actually mean? If you would have asked me to define acid when I was a kid, I would have said it's something that burns people. Now, where did I get the idea that acids were defined as things that burn people? Well, watching TV, of course. When I was a kid, Batman was a TV show, and in this episode right here, the Joker or the Penguin or some villain has captured Batman and Robin, and I don't know how well you can see it, but he's about to lower them into a big smoking vat of sulfuric acid. And it was clear from the TV show that they were going to be burned to death if they got dunked into the sulfuric acid. So I thought, wow, acids are terrible things. They can burn up Batman. Now, of course, I'm sure Batman and Robin escape. They always do. But the point is the show suggested that acids are things that burn people. And here's another thing I remember about acids from being a kid. There was a movie called Westworld, which was about some robot cowboys that start trying to kill people. I think it's actually a TV show these days. But anyway, it was a movie when I was growing up. And at one point, one of these evil cowboy robots was chasing a human, and the guy who was being chased threw some acid in the robot's face, and it was really cool. The acid melted the robot's face right off, and you could see all the wires and gears and computer chips inside his head. It was startling. But again, the point is, I thought, okay, acids are things that burn people. So is that true? Is that actually the definition of an acid? It's something that can burn robots and Batman? No, of course not. Some acids are very strong acids and they can burn you, but most acids are fairly weak and can't burn you at all. In fact, your body makes many natural acids inside your body, and those are actually good for you. Like there are fatty acids and amino acids inside you, and those are normal, healthy parts of your body. Okay, so if the definition of an acid is not it could be something that burns Batman. So what is the actual definition of an acid? Well, you can see it in your lecture outline, or here it is on the computer screen. An acid is defined as any molecule that adds hydrogen ions to a solution, and hydrogen ions are H plus ions, a hydrogen atom with a +1 charge on it. Okay, so I'm going to give you an example of an acid here. We're going to an acid called hydrochloric acid. Let's imagine taking some hydrochloric acid, an hydrochloric acid molecule, and putting it into a solution. This is like underwater. It's in a solution now. Okay, so notice that the hydrochloric acid molecule is a 2-atom molecule. It's made out of one hydrogen atom and one chloride atom. A lot of molecules actually have a hydrogen atom or atoms as part of their molecular structure. So it's not unusual for a molecule to have a hydrogen atom as part of its structure. But what makes a molecule an acid is that hydrogen can break off. And when it does, it becomes a hydrogen ion. So yeah, if a molecule has a hydrogen and that hydrogen can break off and become an H plus, then we say that molecule is an acid. And so that's why hydrochloric acid is an acid. That hydrogen can break off and become an H plus when you put the molecule in solution. Matter of fact, let me just show you that on the cartoon. So you put the hydrochloric acid molecule in solution, in water, and boom, there it goes. The hydrogen broke off. Now, what's left over here, which is a chloride ion that happens to have a negative charge, but don't worry, don't pay too much attention to what's left over. The important point was that the hydrochloric acid molecule that we saw here a second ago was an acid because it can add hydrogen ions to a solution, and how it does that is it breaks off a hydrogen from an atom, and that hydrogen atom that was part of the molecule becomes an H plus, just drifting around on its own in the solution. Now, what we just saw there in the cartoon, the HCl molecule breaking apart to become a hydrogen ion and a chloride ion, we can write that out as a chemical reaction, right? Because a chemical reaction is a way of writing out when molecules get changed. So here's what we just saw written out as a chemical reaction. The HCl molecule turned into a hydrogen ion plus a chloride ion. So I want you to be able to recognize, if I show you a chemical reaction like this, that this is an acid. And here's how you can tell. If you see a chemical reaction and on the product side you see a hydrogen ion, then whatever made that hydrogen ion, on the other side of the arrow, must have been an acid, because that's what acids do, right? They break off a hydrogen atom, and that hydrogen, after it's broken off, becomes an H plus. So anytime you see H plus on the product side, whatever you see on the reactant side where that hydrogen came from, just by definition is an acid, because it can break off a hydrogen and add that hydrogen ion to a solution. Let me give you another example of an acid. So here we are back with our beaker of water, and the acid that we're going to add this time is this one right here. Its molecular formula is H2CO3, but it's called carbonic acid. Okay, so again, the definition of an acid is it's a molecule that can add hydrogen ions to a solution, and how acids do that is they can break off one of their hydrogen atoms, and when that hydrogen atom breaks off, it becomes an H plus that just sort of drifts around in the solution. And so when I click the button, we will see that. Here is the carbonic acid adding a hydrogen ion to the solution. Boom, there it goes. All right, yeah. And so, that just demonstrates that the carbonic acid molecule was an acid because it added a hydrogen ion to solution by breaking off a hydrogen, and that hydrogen became an H plus. If we want to write out what we just saw as a chemical reaction, here it is. The carbonic acid molecule turned into a hydrogen ion, and this other molecule right here. But the point is, if I put this on a quiz or a midterm and said, is this molecule right here an acid? You should say, yes, it is, because over here on the product side, it produced a hydrogen ion, and that's what acids do. They make hydrogen ions in solutions. Good, yes, yeah, so if you see a hydrogen ion on the product side, the molecule that made that hydrogen ion on the reactant side is just by definition an acid. Okay, so hopefully you have a pretty good concept of what acids are, molecules that add hydrogen ions to a solution. There are some molecules that are the exact opposites of acids, and these molecules are called bases. Now, what do I mean when I say a base is the opposite of an acid? Well, bases remove hydrogen ions from solutions, whereas acids add hydrogen ions to solutions. So let's now focus our attention on bases. So here we are, looking at some water right here. So this is a water solution. And so bases by definition are molecules that remove hydrogen ions from a solution. So here's a good example of a base. There's this molecule you see right here. It's called hydroxide ion. The molecule is made out of one oxygen and one hydrogen atom, and the molecule as a whole happens to have a negative ionic charge. It's an anion. Anyway, so bases by definition are molecules that remove hydrogen ions from a solution. And so when I click the button here in a second, you'll see the hydroxide ion doing just that. It's going to remove a hydrogen ion from the solution there. And what does the base do with that hydrogen ion? It just adds it to its own molecular structure. In other words, that hydrogen ion that you're going to see here in a second is just going to become a hydrogen atom that's part of this molecule. Okay, so there's the base in the watery solution. Here comes a hydrogen ion that just happens to be drifting around in that solution. And so what bases do, what their job is to remove hydrogen ions from the solution. So that hydroxide ion base will come over and it will just absorb that hydrogen ion into its own molecule like that. And so, that hydrogen ion is now no longer in the solution. There's no more hydrogen ion. What happened to it? It just became a normal hydrogen atom that's part of this molecule, which I think you recognize as water. But the point is there's now it removed that hydrogen ion that was drifting around freely in the solution, so the hydroxide ion was a base. That's what makes it a base. Now we can write out what we just saw there as a chemical reaction, and here it is. The hydroxide ion reacted with the hydrogen ion in solution, and it turned into a water molecule. So I want you to be able, if I show you a chemical reaction, to recognize whether the molecule is an acid or a base. And how can you tell? Well, if the chemical reaction has a hydrogen ion on the reactant side, whatever molecule that hydrogen ion is reacting with, combining with, that has to be a base, because that's what bases do. They react with, they bind with hydrogen ions. Yeah, so if you see hydrogen ion on the reactant side, the molecule that's with it there on the reactant side that absorbs that hydrogen ion is a base. Let me give you another example of a base. So here we are back with our solution of water here, and the base we're going to see this time is called bicarbonate ion. There it is. So again, remember the definition of bases is they are substances that remove hydrogen ions from a solution. So if a hydrogen ion comes drifting along in the solution, the bicarbonate ion is going to go over to it and absorb that hydrogen ion out of the solution. What happens to the hydrogen ion? It just becomes another hydrogen atom of the molecule, but the point is the base was a base because it removed that hydrogen ion from the solution. Here's what we just saw written out as a chemical reaction. Bicarbonate ion plus the hydrogen ion turned into this molecule here, which is actually carbonic acid. But the point is, how do you recognize that this is a base from the chemical reaction? Well, again, if you see a chemical reaction that has hydrogen ion on the reactant side, the molecule also on the reactant side that it combines with is a base, and so that makes the bicarbonate ion here a base by definition. Okay, so hopefully you have a good concept of what a base is. It's a molecule that removes hydrogen ions from a solution. So let me give you a little drill now. I'm going to show you a chemical reaction, and I want you to tell me whether the molecule you see here in the chemical reaction is an acid or a base. So I'll show you this chemical reaction here, and if I say, look at this molecule right here, which is a phosphate ion, you may recognize that from an earlier lecture. And so looking at this chemical reaction here, is this phosphate ion right here an acid or a base? And the answer is it's a base, because remember, if you see a chemical reaction and you see a hydrogen ion on the reactant side, then whatever molecule that hydrogen ion is combining with, also on the reactant side, is a base, just by definition. And you can see that the two combine, right, because the hydrogen joined onto that PO4, and here it is over here, the hydrogen joined onto the PO4 molecule. Anyway, the phosphate ion that you're seeing here is a base. All right, well, let me show you something that kind of relates to all of this. This is supposed to be a close-up of a glass of water, and these represent some water molecules in that glass of water. Now, obviously, a glass of water would have more than 5 water molecules in it, but let's just go with it. So let's say these represent the water molecules in a glass of pure water. Okay, keep your eye on this water molecule right here. Water molecules can do something called dissociating. And so what does it mean when we say a water molecule dissociates? Well, this is it right here. It means that dissociate means a molecule can break apart. And so when a water molecule dissociates, that hydrogen that was part of the water molecule breaks off and becomes an H+. So let me ask you a quick quiz question. Does that water molecule that we just saw break apart, did it act as an acid or a base? And the answer is it acted as an acid, right? Because it added a hydrogen ion to the solution. It had a hydrogen, and that hydrogen broke off and became an H+, and so that means the water molecule acted as an acid. Okay, so water molecules can act like acids. They can add hydrogen ions to the water. However, whenever a water molecule dissociates like that and puts a hydrogen ion into the water solution, the remaining part of the water, this hydroxide ion, is going to be a base, and bases absorb hydrogen ions. So when I click the button, you'll see that hydroxide ion just takes the hydrogen ion back, and this is going to go back to being a water molecule. And so that's what you'll see when I click the button. But while you're looking at that, also keep your eye on this molecule down here, this water molecule down here. Okay, here we go. That water molecule is coming back together. But look at that. As soon as that one came back together, this water molecule dissociated, adding a hydrogen ion to the solution. And what happens next is, now this one's going to come back together. And as this one's coming back together, keep your eye on this water molecule. There it goes. And so that's the way it works with water molecules. They take turns dissociating. One water molecule will dissociate, adding hydrogen ion to the solution, but very soon that one will come back together, and then another one will then take its turn to break apart and add a hydrogen ion to the solution. So even in a glass of pure water, there is a small hydrogen ion concentration from water molecules dissociating. And it's not a very large amount of hydrogen ions, but nevertheless, there are some hydrogen ions even in a glass of pure water. And you do not need to know the number that I'm about to tell you, but at any given instant in time, only 1 in about 10 million water molecules is dissociated. And so again, that's going to be a very small amount of hydrogen ions in the glass of pure water, but nevertheless, there are some hydrogen ions present even in pure water. Well, keep that concept in your mind that even in water there's a small amount of hydrogen ions, because that's going to be important in the next thing that we're going to talk about, which is called the pH scale. It turns out that the concentration of hydrogen ions in a solution is very important. It can affect, many things. The concentration of hydrogen ions in a solution can. And just to give one example, your blood has a certain hydrogen ion concentration in it. And like so many internal conditions in the body, there's a normal, healthy, homeostatic range of hydrogen ion concentrations in your blood. If your blood, for whatever reason, were to get more hydrogen ions than its normal range, or if your blood were to get fewer hydrogen ions than its normal range, that could cause a lot of problems in your metabolic reactions, and you could potentially even die from having the wrong hydrogen ion concentration in your blood. And just to give one example, if your blood's hydrogen ion concentration gets far enough outside the normal homeostatic range, your nervous system will start to malfunction. And since your nervous system affects the contraction of your muscles for breathing and for your heart beating, you could potentially die. Anyway, the point I'm making here is that the concentration of hydrogen ions in a solution is an important thing. And so scientists invented a way to talk about the hydrogen ion concentration of a solution. And that's what the pH scale is. The pH scale is a way of talking about what the hydrogen ion concentration of a solution is. So now let's talk about the pH scale. So the pH scale, think of it as kind of a number line that goes from 0 to 14. The pH scale goes from 0 to 14. And each solution, each liquid has a certain pH number. A given solution might be pH 4, a different solution might be pH 10.5, another solution might be pH 13.2, a different solution might be pH 7.4, a different solution might be pH 1. Each solution has a certain pH number to it. And again, the pH number of a solution tells you about the hydrogen ion concentration of that solution. That's what the point of the pH scale is to tell you about the hydrogen ion concentration of whatever solution you're talking about. Okay, so let's be a little bit more specific now. So remember what we talked about a moment ago. Even pure water has a certain hydrogen ion concentration to it from those water molecules dissociating. Well, what they did is they said that the concentration of hydrogen ions that you find in pure water, they're going to define that as pH 7. And so, let's use these hydrogen ions right here to represent the hydrogen ion concentration in pure water, and they just called that pH 7 just by definition. So pure water, just by definition, is pH 7. Now notice that pH 7 is right in the middle of the scale. It's not towards the lower numbers, it's not towards the higher numbers, it's kind of neutral. So they also call pH 7 the neutral pH. Okay, so far so good. Well, here's where things get a little bit backwards, so to speak. On the pH scale, the lower the number, pH number, the more hydrogen ions a solution has, and the higher the pH number means the fewer hydrogen ions the solution has. And so I just tell students, memorize this phrase, the pH scale is backwards. The pH scale is backwards, meaning just what I said, that lower numbers on the pH scale mean higher hydrogen ion concentrations in the solution, and higher numbers on the pH scale mean lower hydrogen ion concentrations in the solution. Now you might say, well, why on earth did they make it backwards like that? That's just confusing. Why not have lower numbers mean lower hydrogen ion concentrations and higher numbers be higher hydrogen ion concentrations? Why not do it that way? Wouldn't that be more sensible? Yeah, that would make more sense, but for whatever reason, they made the pH scale backwards. So just accept that lower numbers are higher hydrogen ion concentrations and higher numbers are lower, higher pH numbers are lower hydrogen ion concentrations for the solution. Okay, so any solution that has a pH number less than 7 is called an acidic solution. And so, it may be 6.9, that's an acidic solution, or 6.3, or 5.8, or 5, or 4, or 3.2, or 1, or 0.7. Any pH number below 7 is an acidic pH number, or any solution that has a pH below 7 is an acidic solution. And on the other side right here, any pH number that's above 7, or any solution that has a pH above 7, you call a basic solution, and those are the basic pHs. So for instance, 7.1 is a basic pH, and 7.5 is a basic pH, and 8 is a basic pH, and 11 is a basic pH, and 13.4 is a basic pH, and 14 is a basic pH. Any pH above 7 is a basic pH. Now, why did they get those terms? Well, imagine that you had some pure water. That would be pH 7, right? If you added an acid to that pure water, you'd be adding hydrogen ions to it, right? Because that's what acids do. They add hydrogen ions to solutions. So if you took pure water and added an acid to it, you'd get a pH less than 7, right? Because more hydrogen ions equals lower pH numbers. And so that's why they call these acidic solutions, because acidic pHs, because you would get them by adding acids to pure water. And likewise, if you had pure water, pH 7, and you added a base, that would change the water's pH to something higher than 7. That would change the solution's pH to something higher than 7, right? Because when you add bases, they remove hydrogen ions, so you'd end up with fewer hydrogen ions than pure water, and that's going to give you a pH number above 7. Okay, so pH scale, just to summarize, the pH scale goes from 0 to 14. The pH 7, which is the pH of pure water, is called the neutral pH. Any pH below 7 is called an acidic pH. Any pH above 7 is called a basic pH. And the whole point of the pH scale is it tells you the hydrogen ion concentration of a solution. Now, interestingly, a lot of the foods and drinks that we take in are not neutral. Many of them are acidic and some of them are basic. Here's a little cartoon that shows some of this. And you don't have to memorize what I'm showing here. I do want you to know that pure water's pH is 7, but you don't have to memorize that milk's pH is 6, and you don't have to memorize that coffee's pH is 5, or these food pHs down here. Looks like eggs are pH 8, so they're slightly basic, and baking soda, which they use in a lot of baked goods, I guess that's pH 9. Yes, so many of the foods and beverages that we take in are acidic or basic. Here's my mom and my stepfather, and so they're having Thanksgiving with us. And so the cranberries, those are acidic. There's a type of acid in there called citric acid. Here's the vinegar, that's acidic. There's an acid in there called acetic acid. And here's some wine. There are some acids in there also, but here are some baked goods. These are basic. Um, they have eggs and baking soda in them. All right, so yeah, just so many of the foods and beverages that we take in are acids or bases. Here's the thing, though: your blood has a specific pH range that it's supposed to be. Its healthy, normal, homeostatic pH range for blood is 7.35 to 7.45. And just as I've been talking about, I guess I mentioned this a few minutes ago, if your blood gets outside its normal pH range, you could potentially die. Amongst other reasons, your nervous system would start to malfunction if your blood's pH gets outside the normal pH range of 7.35 to 7.45. And so what I want to mention here is that it seems like a contradiction, right? On one hand, I'm telling you your blood's pH has to stay locked in at this very narrow pH range of 7.35 to 7.45, or you could die. And I'm also telling you that all the time we drink very acidic things or eat very basic things. And so you might say, well, wait a minute, Mr. Edens, if we're constantly taking in acids or bases, the things we eat and drink, how can our blood possibly keep at a steady pH, right? If you think if you ate acidic things like a lemon, those are acids, and so they would give you a lower pH number. And you'd think if you ate basic things like baked goods with eggs or baking soda, that would give you that would shift your pH to something higher than the normal healthy range. So that's my question. I want to examine is how can our blood keep a steady pH within the normal homeostatic healthy range, despite the fact that we eat some pretty strong acids and some pretty strong bases all the time. Well, your body obviously doesn't want your blood to go outside the normal pH range because that would, as I said, that's what would make your nervous system and other organ systems in your body fail, and you could die. So your body has substances in the blood called buffers that help lock in the pH of the blood in the good 7.35 to 7.4 range, despite the fact that you eat these strong acids or strong basic foods all the time. Okay, so yeah, if you look in the web handout, it defines buffer for you. A buffer is a substance that minimizes changes in a solution's pH. In other words, buffers are substances that make a solution resistant to pH change by acids or bases. And that's what your blood wants. That's what your body wants, right? Your body wants your blood to be resistant to pH change so your blood can stay locked in at the good pH range. Okay, so let's discuss in a little bit more detail exactly what are the buffers in our blood that keep our blood in the good pH range of 7.35 to 7.45, despite the fact that we put a lot of acids or bases into our body when we eat and drink things. Okay, well, there are actually many different buffering molecules in the blood, but the two major buffers in the blood are called carbonic acid and bicarbonate ion. So imagine we're going to zoom in on a small part of the blood. Imagine somehow we can just zoom in on one drop of blood. And that's why the background color in this slide is red. Imagine we're zooming in on just one tiny, tiny drop of blood. Okay, and so in this little close-up of the blood that we're seeing, let's say these three hydrogen ions represent the proper amount of hydrogen ions for the blood. In other words, these three hydrogen ions represent the amount of hydrogen ions to keep the blood in the 7.35 to 7.45 pH region. Or said another way, the body's goal is to keep just three hydrogen ions in this part of the blood that we're seeing here. Okay, so in addition to the hydrogen ions, notice that the blood also contains carbonic acid molecules, which are the ones over here, and the blood also contains bicarbonate ions, which are these molecules over here. And again, the job of the bicarbonate ions and the carbonic acid molecules is to be buffers. Their job is to prevent pH changes of the blood, or in this terms of this cartoon, their goal is to help keep the three hydrogen ions that you see there. Okay, so let's now explore a scenario that could potentially change the blood's pH. Let's say you drink some acidic beverage. Maybe you have a bunch of lemonade. Well, yeah, lemonade is acidic, and acids add hydrogen ions to a solution. So when you drink that lemonade, you're adding hydrogen ions to your body. And so here comes one of those hydrogen ions from the lemonade into your bloodstream. Okay, so let me just stop right here. The number of hydrogen ions in your blood has momentarily increased, right? So is your blood's pH in danger of going above the normal range or below the normal range when you add hydrogen ions, extra hydrogen ions to it? And the answer is below the normal range, right? Because remember, the pH scale is backwards, and so the higher the hydrogen ion concentration, the lower the pH number. And so if extra hydrogen ions come into your blood, you're in danger of getting your blood pH below the normal pH range of blood. Okay, anyway, so this extra hydrogen ion has come into your blood, in this case from the lemonade that you've been drinking, and your body wants to keep you in the normal pH range of 7.35 to 7.45, which, in terms of our cartoon here, means the body only wants 3 of these hydrogen ions, not 4. So how does the body get rid of this extra hydrogen ion? The answer is the bicarbonate ions. Bicarbonate ions are bases, and bases absorb hydrogen ions. So the job of the bicarbonate ions in your blood is to absorb any excess hydrogen ions that come into the blood. And that's exactly what we're going to see that bicarbonate ion do. It intercepts any extra hydrogen ions that come into your blood. And, bicarbonate ion is a base, so it just absorbs them. What happened to that hydrogen ion? Well, it just became another hydrogen atom that's part of this molecule here. But the point is it removed the hydrogen ion from the blood. And so that's what bicarbonate ions do. They buffer your blood by absorbing any extra hydrogen ions that come into your blood. Now let's look at the opposite scenario. Let's say that your blood's normal pH, these 3 hydrogen ions, what happened is you've lost one of those hydrogen ions. So how did your blood lose hydrogen ions? Well, maybe you were vomiting. Excessive vomiting can cause the body to lose hydrogen ions, and so that would eventually make the blood lose hydrogen ions. So another question, let's stop the action here for a second. When your blood loses hydrogen ions, is your blood's pH in danger of going above or below the normal pH range for the blood? And the answer is above, right? Because the pH scale is backwards, and so if you remove hydrogen ions from a solution, its pH goes up. And so, yeah, if you've lost hydrogen ions from your blood is in danger of going above the normal pH range for blood. Okay, so let's say something like that happens. So your blood's pH is starting to go above the normal range. The buffering molecule in this case is the carbonic acids. Carbonic acids are acids, and remember, acids add hydrogen ions to solutions. And so if your blood starts losing some hydrogen ions, the carbonic acids will add hydrogen ions to the blood to get you back into the normal pH range. And here we go, is that carbonic acid, and it's an acid, so it adds hydrogen ions to the solution. And so now notice that thanks to that carbonic acid, your blood has been restored to the proper pH range. Good. So summarizing all this, the buffers are substances that keep a solution locked in at its proper pH range. Your blood has buffers to help keep the blood locked in at the proper pH range of 7.35 to 7.45, and the major buffers in your blood are carbonic acid molecules and bicarbonate ions. The bicarbonate ions buffer your blood by removing any extra hydrogen ions that enter the blood, and the carbonic acid molecules buffer your blood by adding any hydrogen ions that you need if you've lost hydrogen ions from your blood. Okay, that brings us to the end of this lecture on water. As usual, I recommend that you now try to memorize your notes, but then close your notes and see if you can do the water review questions from the course website. Okay, thank you. I will see you in the next lecture.